Hi students. So our next topic is enzymes, which is actually very important topic. Not to mention that we are covering the following syllabus: Cambridge Checkpoint, IGCSE, GCSE and AQA Biology. So before going ahead, let me introduce you what are enzymes. Okay, many of our staple foods such as rice, potato, pasta or bread contain large quantities of starch, isn't it? You eat all these. Then take a mouthful of one of these plain without anything else. And at first there is not much taste. Have you observed? Okay, so if you continue chewing on it for a few minutes to mix it with saliva and reduce it to a slush, you will find it starts to taste sweeter, isn't it? So this is because saliva has enzymes that start to break down the starch into smaller sweet sugar molecules. Okay, so enzymes are essential in digestion. Why they are essential? Because it has to break down the large molecules in our food into molecules small enough for diffusion through the cells of the gut wall and into our bodies. So let's move on. As you can see in the heading, I have put biological catalyst. Okay, so do remember that enzymes are biological catalysts. In the next slide, I'm going to tell you what is catalyst. A catalyst is a substance that speeds up the rate of a chemical reaction but if not itself changed by the reaction. And why do we call biological catalyst? Because it is present in living cells, biology, isn't it? So catalysts that control metabolic reactions are enzymes because they work in living cells. That's why they are called biological catalysts. Enzymes help cells carry out all the life processes quickly. Without enzymes, these metabolic reactions would happen too slowly for life to carry on. Okay, so we have to remember that enzymes are biological catalysts. Okay, so here I have just put up one example of breaking down of hydrogen peroxide. Hydrogen peroxide, which formula is H2O2. Okay, so catalyst will break it down into manganese oxide will react as catalyst. It will break it down into water and oxygen. Manganese oxide is a catalyst. What does it mean? As you can see here in the equation, it is not broken down. Means it remain unused at the end of the reaction and it can be reused. So same goes to enzymes. Look. Okay, so this is our here the escaping oxygen causes the foaming. Have you ever tried this hydrogen peroxide? You can try it at home. Put some foam in it, okay, like soap and water. That will actually make the foam and you can see the oxygen gas escaping as the foam. Okay, let's move on. Enzymes act as biological catalysts. I have explained to you what is biological catalyst. They occur inside cells or secreted by the living cells. That is why. And catalase is the enzyme that catalyzes the breakdown of hydrogen peroxide. Catalase naturally, do you know where it is present? It is present in potatoes as well. Okay, so catalase. As you can see that it will break it down. Now next, enzymes are proteins. Yes, remember. So one of the, how will we write the definition in the exams? Enzymes are biological catalysts. Then second mark, enzymes are proteins. Okay, enzymes are proteins. Proteins are long molecules that are folded into a specific shape. Here, I show you different shapes. This is catalase, amylase, pepsin and trypsin. And you are going to learn more about it because enzymes are also specific. Okay, what does it mean? It means each enzyme only works with one substance or a similar group of substance called its substrate. Like for example, here catalase is a type of carbohydrate enzyme that will work for the digestion of starch molecules, okay, into simple sugar cells, into simple sugar, I mean. Okay, then similarly amylase also a carbohydrate. Pepsin is for proteins. This is a protease, okay, and that break down proteins into smaller units. Trypsin is also a protease. So catalase and amylase will work for carbohydrates, while pepsin and trypsin will work for proteins. So here in this lesson what we have learned, that enzymes are biological catalysts, okay, they are proteins. Now so here the part of an enzyme where the reaction occurs is known as the active site. Okay, so as you can see, enzyme, it has an active site. As we have just mentioned that enzymes are specific. So means this active site will accept only specific substrate. Substrate is going to be the molecule. Okay, like starch, proteins, understand? So see here in the animation, these are the substrate molecule, and active site, the site on the enzyme where the reaction occurs. Now you see here, okay, so products will be formed, okay, and what will happen then, you know, they will leave, okay, the enzymes means it can be reused. Look, yes, okay, it is leaving, then you know the next substrate coming in and it will again be broken down. Okay, so what we have learned, that enzymes, they can be reused. Okay, product will be formed. Enzymes have specific site, active site. In the exam we are going to use these keywords. Substrates are the molecules need to be broken down, like for example starch, proteins. Okay, and once you know enzyme carry out its function, the product can leave the enzyme and enzyme can be reused. So here we end this lecture and I'll take you to the second lecture of enzyme. See you there. Bye.